Small intestinal bacterial overgrowth, intestinal methanogen overgrowth, and intestinal sulfide overproduction are three distinct gastrointestinal conditions, each characterized by a unique imbalance in the gut microbiome. While the identification of specific microorganisms associated with SIBO, EMO, and ESO has advanced our understanding of these conditions and the development of targeted treatments, no studies have explored how each condition affects the host's small bowel gene expression. Gene expression is the process by which the genetic instructions in your DNA are used to make a messenger RNA molecule, which is then translated by the cell to perform specific tasks like the creation of proteins. Because the microbiome can significantly influence a host's gene expression, next-generation RNA sequencing, transcriptomics, can offer valuable insights into the mechanisms underlying health and disease states. So, how do these gastrointestinal conditions affect small bowel gene expression? In this study, we aim to explore changes in human small bowel transcriptomics in SIBO, EMO, and/or ESO, and to investigate if changes in ESO reflect those seen in rat models gavaged with known hydrogen sulfide producers. Researchers selected samples from 85 participants enrolled in the ReIMAGINE study, who had the diagnosis of SIBO and/or EMO and/or ESO. The ReIMAGINE study is an ongoing investigation aimed at understanding the role of the small intestinal microbiome in human health and disease, utilizing samples obtained during standard of care upper endoscopy. All participants underwent fasting breath testing to measure methane and hydrogen sulfide. Small bowel biopsy samples from every participant underwent transcriptomic analysis to identify differentially expressed genes and altered biological pathways. To validate the human findings, researchers also examined rats gavaged with the H2S producing bacteria Fusobacterium barium or the Sulfovibrio piger, alongside rats gavaged with one of two Escherichia coli strains as controls. Researchers then identified similar changes between the rat small bowel transcriptomic data and ESO. And here are the results. Please note that when conditions are analysed exclusively, they will be distinguished with only beside the name. Overall, subjects with ESO only exhibited more significant differences in gene expression than subjects with only SIBO or EMO. Researchers found SIBO versus non-SIBO showed no differences in overall transcriptomics profiles, but there was a downregulation in the gene expression of SLC30A2. When adjusted for age, none of the genes were differentially expressed. In SIBO only versus subjects without ESO, EMO, or SIBO, the NNN group, no differences in demographics or overall transcriptomics profiles were found, but 3 DEGs were identified as downregulated. Comparing EMO versus non-EMO, there was no difference in demographics or overall transcriptomics profiles. Also, no genes were differentially expressed. When subjects with EMO only were compared to NNN, no differences in demographics or overall transcriptomics profiles were found, but 5 DEGs were identified. 4 were downregulated and 1 was upregulated. Now looking at ESO, researchers first compared subjects with high ESO to non-ESO subjects. They detected significant differences and identified 233 DEGs. The main biological processes involved were RNA splicing and response to oxidative stress. The same analysis was performed for subjects with lower H2S levels. Transcriptomic profiles remained significantly different between ESO 1.5 and non-ESO subjects, with 923 GEGs identified. The main biological processes were response to oxygen-containing compound and mitochondrial ATP synthesis. When all ESO subjects were combined and compared with non-ESO subjects, researchers again found significant transcriptomic differences and identified 643 DEGs. The main biological processes were mitochondrial ATP synthesis and cellular respiration. Subjects with ESO only were compared to subjects in the NNN group. While there were no overall differences in transcriptomics profiles, 17 DEGs were identified. The main biological processes involved were response to stress and immune response. For ESO only 1.5 versus NNN, overall transcriptomics profiles trended towards being significantly different, and 270 DEGs were identified. The main biological processes were activation of mitochondrial respiration and inhibition of immune response. In addition, an endogenous H2S producing enzyme, CTH, was found to be a downregulated gene, suggesting the cell is responding to toxic levels of H2S in the environment. When all ESO-only subjects were compared to NNN, no overall differences were found between the groups, but 132 DEGs were identified. For the main biological processes, there was decreased expression of immune response genes and increased expression of genes associated with mitochondrial ATP production. The downregulation of CTH in both ESO-only comparisons suggests that cellular endogenous H2S production is decreased. Importantly, SIBO-only, EMO-only, and ESO-only comparisons did not share a single common DEG. Now, when examining rats scavenged with known H2S producers, Fusobacterium varium or Desulfovibrio piger, and rats scavenged with one of two Escherichia coli strains, were used to explore their effects on rat small bowel transcriptomics. F. varium gavage rats showed significant differences in overall transcriptomics profiles versus controls, and had 1,244 DEGs, including genes associated with mechanisms found in humans, such as redox balance, water homeostasis, gut motility, and IBS-associated pathways. Comparing deep hydrogavaged animals to controls, no DEGs were identified. Lastly, rats were gavaged with E. coli BL21 or E. coli K12, a facultative H2S producer, and were compared to controls. Overall differences in the transcriptomics profiles of E. coli K12 gavage rats versus controls were close to significant, and 137 DEGs were identified, most of which were downregulated and associated with oxidative phosphorylation, ATP synthesis, iron regulation, and response to hydrogen peroxide. E. coli BL21, which shares more than 99% sequence identity over approximately 92% of the E. coli K12 genome, did not show overall differences in transcriptomics profiles versus controls, and had 13 DEGs which were not associated with the main mechanisms found in E. coli K12 and ESO. E. coli K12 cultures were H2S positive, suggesting that H2S production may be involved in differences in gene expression in rats and humans. In summary, subjects with EMO and SIBO had some differences in host gene expression, while subjects with ESO displayed the most distinct and extensive differences in small bowel gene expression. Hydrogen sulfide levels of 1.5 ppm or greater were associated with widespread alterations in genes involved in mitochondrial ATP synthesis, redox balance, gut motility and pain, antimicrobial, ferroptosis, and water homeostasis pathways. Moreover, the rat experiments closely mirrored the human ESO findings, demonstrating alterations in genes related to mitochondrial ATP synthesis, redox balance, gut motility and pain, water homeostasis, and IBSD-associated mechanisms. Together, these findings suggest that ESO represents a biologically distinct condition with unique effects on host transcriptomics and may require different therapeutic strategies than SIBO or EMO. SIBO-only, EMO-only, and ESO-only comparisons did not share a single common DEG. Thus, they will potentially benefit from different treatment approaches.